Hello. The DNA of all cellular forms is double-stranded, and all life is the common property of all life is having DNA or the genetic information. And the important part of genetic information is the ability to manifest the whole physical form of life and also replication, the propagation itself, which is the basis for reproduction as well. And all biological information is present in the form of sequences, the nitrogenous bases that are present here. And that is the sequence. The sequence of these nitrogenous bases is the information in which is stored in the genome. The problem is that as a genetic information or a genome, the DNA has to manifest all the cause phenotype and also be able to replicate. That means the information should be read, and that means the DNA is both the strands are closed upon themselves, they should open up, and then if they open up, there is a problem, that is, they are vulnerable to be damaged. But if they are closed, then they cannot be read. So these are some of the problems that DNA as genome faces. And there are fantastic mechanisms of how the evolution has come up to solve these logistical problems. So DNA should denature, that is separate into two strands, but also renature, be stable. For stability, you need to be in double-stranded stage. For revealing the information or copying the information or transcribing or manifesting the features, you need to open it up, at least transiently. And this also has a lot of problems with it, as we discussed in cases of topology, the supercoiling. Say, for example, we have a DNA molecule fixed at both the ends like this, and we have the DNA molecule which is having twists. This is 1 twist, 2, 3, 4, 5, 6, 7, 8. There are 8 twists here. And if we were to put in a stick there and try to push it to one of the sides, then there are more twists in this location compared to in this much. Previously, we had about, say, 1 twist per unit length. And now we have compressed them. All the twists are here, which were otherwise spread until here. So here we are having superhelical density. We have more number of twists than there are. That is what is supercoiling. And here it is forming positive supercoils because there are more number of twists than the short length. So if we were to keep on moving, there will be a point where there will not be any further, we cannot move it without breaking the DNA. So if we still forced to do that, then the DNA the backbone will break. So to relieve that stress, they do form supercoils, something like this. There are supercoilings. On this side, we have positive supercoiling, and on this side, we have negative supercoiling. And both of them can form right by twisting upon themselves. To relieve the tension, the backbone, the phosphate backbone, is experiencing because of this supercoiling. How does DNA relieve it? How does DNA or the genome, how does it manage to allow replication to happen? How does it allow transcription to happen? And that is what we will discuss now. I am Dr. BCMR, Assistant Professor at Shastra. We'll talk about some of the beautiful enzymes that play or kind of manipulate the topological structure or topology of the DNA or in the form of genome. So those are called as topoisomerases. If this is a replication fork, which is where the helicase is the enzyme that is opening or denaturing the two strands so that they are separated, and that will be stabilized by single strand binding proteins and other things. On the upstream of the replication fork is moving in this direction, and upstream of it, that is where the positive supercoiling is building, and this DNA would have been bending upon itself in such fashion. How does it relieve the tension, and that is by topoisomerases. And in DNA, in E. coli or in bacteria, we call it as DNA gyrase. DNA gyrase is an enzyme. It is a multimeric protein with two subunits, gyre A and gyre B. And what it does is something like this. So if this is a DNA molecule or double-stranded DNA, and one DNA, it is looped upon itself, something like this. What it does is it will make a cut in one of the DNA, and then it brings these two strands upwards, or send this strand, the DNA molecule, double-stranded one, below it, and then seals it up. So upon sealing, it should look something like this. If we can make it up, this is how it would have changed. So it has taken a the double-stranded DNA, and pushed it by cutting the bottom DNA molecule. It pushed it below. So it would have reduced the twist by a number of 2. So the enzyme itself looks something like this. Assume this is where one of the DNA molecule is bound, and this is another looping one. It would have taken one of these strands, one of the DNA molecules, and put it here, and then it will break the other DNA here like this, and then pushes the DNA molecule here, and then it will seal up. It works. For visualization, that should be a nice way of thinking. When we try to use these gestures using fingers, it is fantastic in terms of structural biology, it is a good way of appreciating how proteins work. They work just like how we would have solved. And how did they come about is a fantastic way of thought process too. How did evolution come up with these? And this is all in proteins, that is the structural information. It's not just about how the protein, what is the primary sequence. It's not just about how it should be, the conformation itself, but it's about how the conformation changes based on the ligands that are bound, or the substrates that are bound, or the surrounding conditions. That is also the part of genetic information. The behavior of the protein based on its structure and activity of the functional groups in the active sites, everything comes under the processes of life, or that is where how life manifests. Okay, there are also other topoisomerases, topoisomerases 1 and topoisomerases 2. What we discussed about DNA gyrase, it's an example of topoisomerase 2. We will see the differences shortly. First, the topoisomerase 1 is something like this. These are two strands of DNA. Denaturing can occur. And here, what topoisomerase 1 does is it makes a cut in one of the strands, not both the strands like the DNA gyrase does. It cuts, it twists, and then joins them together. And now we have reduction in one twist, right? Because we cut it up and then we joined it here. So there is a reduction of twist 1. So there are different models about it, but essentially the point is they make a cut and they form this protein DNA junction or linkage, and then they pass the other strand and reseal it. And that is how topoisomerases 1 works. So topoisomerases are important for several functions within the cell. During replication, when DNA polymerase is moving, you have supercoils that need to be relaxed, and positive supercoils are relaxed, and which means you are actually inducing kind of negative supercoiling. And there are also other functions, such as if it is a bacterial replication, they are circular. The genomes are circular, and upon replication you would end up, the same is the case with the plasmids too. They would end up with concatenated DNA molecules like this, and we would need double-stranded breaking topoisomerases, that is topoisomerases 2, to break them up and seal them back like this. And so Topoisomerases play an important functions. They are essential for replication and many other aspects that should, such as transcription and even in recombination as well. Let's quickly have a difference, try to differentiate between what are topoisomerases 1 and topoisomerases 2. So topoisomerases, they make single strand breaks, and topoisomerases 2, they make double strand breaks. It's easy to remember that way. And topoisomerases 1 are not, they do not require any energy. They use, when they break the phosphate backbone, they use the same energy to seal it up. They're conservative in that matter. Whereas topoisomerases 2, they require ATP for their functions. So the ATP would be required to change the conformations and many other stuff for the enzymes. So one another important one that we need to remember is topoisomerases 1 reduces the supercoiling in steps of 1, whereas topoisomerase 2 reduces in steps of 2. I hope these are clear. Thank you very much.